Thank you everyone for coming. It's my great pleasure to present my work done at Beckman Institute, work with Professor Gabriel Popescu. And we are from the Quantitative Light Imaging, which is actually inside this building. So at the beginning, I would like to show you this movie so you get a feeling what kind of questions we are like to addressing. So this is the live imaging of the neuron cells. We can see there are a lot of dynamics happen here, and it's quite complex. For example, these vesicles here, they are busy traveling back and forth in these highways, and the membrane is keeping fluctuation, and the microtubule buckling and the philopodia is do the bending motion. So this is quite a complex system. But physically, we can decouple this dynamics into two physical modes. One is random motion, the other is determinist motion. Random is passive and determinist is active motion. It consumes ATP. Okay. So this is the Brown first observed this phenomenon, the random motion, under the microscopy and published the first paper. But the first theory is proposed by Albert Einstein, and he found out that the correlation is due to the random motion is due to the correlation with many molecules, and by tracking the trajectories of these particles, and we can calculate the mean square displacement. And if we plot the mean square displacement versus the delay of the time, from the loop we can get the diffusion coefficient, which is associated with the viscosity of the medium and the particle size. And actually, interestingly, this paper is Einstein most excited. The study paper has cited most, and because it's easy to understand because the random motion impacts every aspect of our daily life. But inside the living cell, if for the object want to move from point A to point B, the free lunge is not enough, so they need, for example, this calcium motor, they're walking along the microtubule and carry the vesicles you see in the top of it. And this is determinist motion, and which need to consume ATP. So the conventional method, the traditional method to study this dynamics is tracking the particles. So for example, this is some particles this is heart beating cells. I track their trajectories. But particle tracking is not always easy. For example, for this case, this continued distribution particle is very difficult. So what's our first question is if we can propose a method which is free of particle tracking. So and we think of if the light object pass through the object, this wavefront, the wavefront deformation is can be calculated by the phase shift, and which is associated with the reflection index contrast and also the thickness of the object. And the fluctuation of the phase has two contributions. One is the out-of-plane thickness fluctuation, and one is the in-plane reflection index fluctuation. So, and the reflection index fluctuation is associated with the mass transport in plane. And we propose this dispersion relation to describe the mass transport. And this dispersion has three components: directed motion, and diffusion, and train diffusion. Here, Q is the spatial mode, spatial frequency, and gamma is the time bandwidth. So this one, so directed motion can be described by the first polo, and this is quadratic is for diffusion, and the cubic for the train diffusion. And we name it dispersion-relation phase spectroscopy. So the next question is, how can we measure the phase? Since the live cell is very transparent under the bright field microscopy, and the laser has the problem. Okay, laser can see this, but laser, due to the speckle, the high coherence, there are a lot of speckle. The phase contrast greatly enhance the contrast, but it cannot directly get the phase. So in our lab, we developed the called spatial light interference microscopy, and we can see it's quantitative and it's dynamic, and due to the white light source, it's speckle-free. So this is our setup. So this is conventional phase contrast microscopy, which won the Nobel Prize, and our system is the slim modular, which has auto modular. So compare the phase contrast microscopy, which create a pi/2 phase shift between the scattered light and unscattered light. So this modular, the slim modular, creates 3 additional phase shift and record each images. And combine this image can create this this quantitative phase image, and this color band means the optical path length in nanometer scale, and this is label-free. So we use the particle bead to test the DPS as proof in INSPR. And here we can charge hundreds of particles and calculate this mean square displacement, get the diffusion coefficient. And this one is we apply our method DPS, and this is log scale by plot the dispersion curve, gamma versus Q, this quadratic, and by fitting, which we got the diffusion coefficient agree quite well with this gold standard. And then we apply the method in the live cell. This is 2 microglia cell. How we do that? Basically, we do 3D Fourier transform. So first the 2D spatial Fourier transform, then the next 1D temporal Fourier transform. So and we plotted the curve. We found that there are 2 regions, 2 distinct. And for this one, the high Q, which remember this spatial frequency 1/L distance. So this is at a local scale, and we found at the local scale is diffusion dominant. But if you for the high Q, for the low Q part, which means the high spatial scale, so determines motion dominant. And then this is our first movie, the neuron one, and we found that the dispersion map is not isotropic anymore. And also we got at this very high Q region, we found the cubic polymer dependence, which is about the philopodia, the bending motion. So we can quantify the intracellular transport inside the living cells. We can get the diffusion coefficient, we can get the velocity. And for this, we can see the membrane is more diffusive. And here, if from transport from the nuclear to the membrane to the periphery of the cell is combined 2. But for the dendrite of the neuron cell, it's determined motion mostly. Yeah. And we can straighten out these dendrites, we quantify it, we can get the velocity distribution is about 2 to 4 nanometer per second. And we also can calculate the correlation at each pixel. So this one, the top row is for the normal glial cell, and this we use the cytotine to block the actin polymerization, and we calculate the correlation time. So this is for the low Q part, and this is for IQ part, high Q. We found that the high Q is not defected by this the actin block, but for the low Q, the larger scale, we can see there is a lot of different. So if we want to do the serious biological study, we need a specificity. So we need to know which component is actually moving. So we need a fluorescence. Inspired by the FCS, fluorescent correlation spectroscopy, which make use of the fluorescence intense fluctuation inside this confocal volume to study the particle movements, conformation damage, changes and chemical reactions. We propose the dispersion-relation fluorescence spectroscopy, which share the same spirit of the DPS. And see, this is the fibroblast cell, and with these are the green, these are the actins labeled with the GIP, and we found along this fiber direction is mostly determined motion, but the perpendicular direction is more diffusive. So this is our recent work. Do I have time?
Yeah. Another couple of minutes.
Okay. Okay. So this one this is the case we blocked the microtubule processing with the knockdown, and the second one we blocked the actin with the cyto D. And we found with this drug treatment, the determinism motion later will become diffusive. But the interesting we observed is here. So after the determinism motion become more diffusive, become disrupted. But after a while, about 20 minutes, seems returned back to the determinism motion. So we puzzled about that. So our hypothesis is maybe this blocked cyto D segment, they fall into the microtubule, and so the microtubule can carry them more deterministically. So we do this control experiment. We block the actin first, and then we block the microtubule, and we found after the determinism motion, after you return back the impulse motion, and if we block the microtubule, so finally it becomes the purely diffusive. So this is our summary. The quantum phase image enables the label-free dynamic studies, and the DPS dispersion-relation phase spectroscopy, no tracking, and we can study dynamics along in the broader spatial and temporal scale. And the intracellular transport is locally diffusive and globally deterministic. And the cyto D and blobbing study affect the larger scales. So thank you, our QUB lab and our collaborator. Most of the work is done. The collaboration with Master's lab, and also the fluorescence is more with Peter Wang, which is also Beckman Institute affiliates. Okay. Thank you.
Well, thank you very much. Any questions? Come on. Yeah, in the back.
Thanks for the presentation. It was really good, really interesting. Do you guys have a way of detecting which specific particles are moving?
Which is specific? So basically that's why we developed the DFS, dispersion-relation fluorescence spectroscopy. So with the fluor attached to the components you are interested, so we are able to.
Yeah. Another question? No? Well, thank you once more.